So to gather our plankton samples, I use this plankton tow net, which is a very fine mesh and then ends in this bottle right here. And on the side of this bottle, there's a 120 micron mesh. So this allows water to escape but retains any particles larger than 120 microns. So I collected it in there. I transferred it to this vial. There's a teeny world in there. And then I take little subsamples of it and I put it into a petri dish like this. I turn on the lights, and then I'm using a compound microscope, or sorry, a dissecting microscope here with my camera attached to it. And then when I want to really zoom in, I bring it over to my compound microscope, which will go up to 200 times magnification. Whereas this one's only going to, somewhere between 10 and 75 times magnification. So this is our sample. You can see, because I took my sample right next to a dock, there's a lot of chunky stuff that's not strictly part of the plankton, more of a fouling community. We'll get to that stuff at the end, but I'm going to start with sort of a tour of some of the stuff I found in the sample. I'll kind of start small and work my way towards larger. Alright, now we can look at a couple different types of diatoms. So diatoms are phytoplankton that build their tests, instead of like most organisms we're familiar with that make shells out of calcium carbonate, they actually build them out of silicon or silica. So basically they have glass shells. So you can see these little balls here. Some of those are diatoms, come in tons of different shapes. So those are going to be some relatively large diatoms. You can see there's some bigger ones around. But then you have this little teeny one that's sort of crawling around. There you go, it's just crawled into one of the balls, bumped right into it. Oh, there's one. That's a nice square one. So here's a perfectly square diatom. I think that's beautiful to see such symmetry in nature. There's a lot of detail when you get down to the small size. Well, here's a good shot of some diatoms growing attached to some algae that was floating around the sample. You can see those two green lines shooting off to the left. Here we've got a colony of diatoms on the right, and then on the left, this wriggling thing is, I believe, a larval tunicate. Something that looks like a tadpole for now, but will eventually turn into something very strange and alien looking. Then down below, there's a foram. So this is another single-celled plankton. It's an amoeboid, and it builds these calcium carbonate spiral shells. Alright, check this out. This is that vase-shaped organism, sort of in the middle of the screen there. You can see, oh, there goes a copepod. That is actually a gigantic single-celled ciliate. So it's got a bunch of cilia at the, if you look down in the screen there, towards the opening of this vase shape, it's where it's got a bunch of cilia that brings water in to basically its mouth, functionally. And this is, for a single-celled organism, that is quite large, because you can see it's almost the same size as that, little copepod that's swimming around. All right, now we're moving into the realm of the multicellular animals. So a lot of what's going to form the zooplankton are going to be larval versions of animals that we're more familiar with. So for example, even something like a tuna fish, which might weigh hundreds of pounds as an adult, that's going to start out as a teeny little larval fish that's maybe only 2 millimeters long. And what we have here is a larval polychaete, a polytrochophore is what that's called. Organisms of this scale exist with really low Reynolds numbers, which means they live in a world dominated by viscosity. So you can see this polytrochophore beating its cilia, which creates a current of water around it. And these spherical phytoplankton are caught up in that current. So as that polytrochophore continues to grow, it'll eventually metamorphose, add subsegments, and continue growing into an adult polychaete. So here's an intermediate stage, I'm pretty sure, not totally sure what I'm looking at here. You can see it's added a few segments, and you can see the bristles, or the chaetes, coming off the underside of this thing. Very strange. Here's a
further developed intermediate stage, or at least it looks more like what I recognize as a sort of normal polychaete. You can see it's got a bunch of bristles. You can see its eyes coming down to the lower right. And then eventually that might grow into something like this, a full-grown polychaete. Here you can see one moving in amongst all the sponge spicules and detritus in the sample. But of course, those polytrochophores could turn into something else that looks like this. Adult polychaetes take all sorts of different forms, so it could end up developing into a sessile polychaete like this feather duster worm here, which lives in a calcareous tube that it builds. Another possibility is that the polytrochophores could develop into a polychaete that looks like this as an adult. This is a terabellid worm, or a spaghetti worm as they're known, and you can probably figure out why they're called that based on this image. But these things feed by sticking their gooey, long tentacles and pushing them out into the environment around them to collect detritus. Not all the worms you're going to see in the plankton samples will actually be polychaete worms. There's a lot of different things that qualify as worms by being long and having no limbs. So here you can see a nematode worm squirming around from a completely different phylum. All right, here we see some different larval zooplankton. On the left, you can see a cluster of some kind of eggs. I don't know exactly what kind. But what I see in the middle is a veliger. So veliger is the name for larvae of mollusks. And this is a gastropod veliger, which means basically a baby snail. And you can see that spiraled snail shape. And if you look really close right now, you can see the cilia beating inside that teeny snail shape, which is how it brings water and food to its body.
So we've got a nice contrast here. So right here, this looks like a bivalve clam, but it's actually the exoskeleton of an ostracod, which is a kind of crustacean that lives in a two-valve shell. But right below it here, we have the veliger, which again is the larval form of a mollusk. We have the veliger of a bivalve. So you can see this is someday going to turn into some sort of clam. You can see little pieces of its foot coming out of the shell there. This thing in the middle here, I believe, is a larval cnidarian, so either a larval hydroid or sea anemone or something like that. You can see it's got a bunch of tentacles coming off, facing away from us.
Crustaceans often dominate zooplankton samples. Here you can see a larval barnacle, or Anopolis. You can see that barnacles start out as free-living organisms only after they go through 5 Anopolis stages and a cyprid stage that they settle down, find a rock to stick to, and then build calcareous plates around themselves. If we zoom out and look at our sample from the perspective of a close naked eye, you can see all these little teeny things squiggling around, and those are mostly going to be copepods, which often dominate the sample. I've zoomed in a little bit. Now you can see this copepod up close a little bit better. And you can see one of its defining characteristics, which is that single red eye right in the middle of the front of its face. And copepod translates to oar feet in Latin. Copepods use those teeny little oar feet to move around in the water. And again, because they're such small organisms existing at really low Reynolds numbers, the water to them feels like molasses would to us if we were swimming around in that. So you can see how they move in lurches and jumps. It's not smooth movement. They don't carry any momentum forward. So they live in a world that's dominated by viscosity, not inertia. All right, I found a copepod that's still alive, but probably not for very long. But because it's sitting still, you can get a close look at that single eye between the antennae and the tail pulsing. The zooplankton includes lots of larger crustaceans as well. So things like this amphipod here might feed upon some of the smaller zooplankton we've been looking at, and also detritus and phytoplankton. Here's a larger amphipod. This is actually a benthic species, but it showed up in the sample, so I thought I'd include it.
Alright, here you can see a mysid shrimp. This is one of the larger things that we might consider zooplankton in this sample. They're going to cruise around eating a lot of the smaller zooplankton that we'll look at later on here. And you can see that it's furiously pulsing its pleopods, those teeny little legs, to keep water moving past its gills. And you can see through, so you can kind of see like right into its belly and its brain, which I think is pretty neat. Alright, I zoomed in even further on the mysid, just because I wanted to get a close-up of the tail. Somewhere in their tail they have statocysts, which give them a sense of balance. I'm going to move up the body. I'll have to move the plane of focus as I do that. Oh yeah. Oh, you can see its heartbeat. Look at its brain. There's its eyes.
So to understand the relative abundance of these organisms, we need to count them. Obviously, there's too many to count all of them, so we're going to use a gridded petri dish to count subsamples of our sample. So here you can see a bunch of 2 millimeter by 2 millimeter grids, and we'll zoom in on one and count.
So when you're counting organisms, it's much easier. It's more beautiful when you're looking at live ones, but it's a lot easier to get accurate counts using dead ones. So I'm going to switch over to some samples from the previous year, which are preserved in ethanol. Ooh, what was that? All right, and then in this grid, I've got 1, 2, 3, 4, 5, 6, 7 copepod, 8 copepods, and a ciliate. In this cell, we've got 1, 2, 3, 4, 5, 6, 7 copepods, at least 2 diatoms shaped like pens, and 1 veliger, I believe, of some sort of gastropod.
So normally we would have you all in the lab going for your own samples gathered from Mission Bay, but we can't do that this year. So I'm going to give you data from previous years, and the rest of this video is just other cool things that ended up in my sample that aren't actually plankton.
Alright, I brought some of these water sporia bryozoans in under the compound scope, and now you can really see what they look like. So they're all packed in there. Each of those dark blobs is the operculum out of which the feeding lophophore will come out. And each one of these is connected to its neighbors. So there's this like network of tubes where they can all share resources. So they end up, oh, there's one's lophophore's coming out. Oh, cool. So that's how it feeds. Look at that. You can see how the operculum door opened up and it sticks its feeding tentacles out. Wow. Oh, there it goes. Closing the door. So yeah. Alright, this is my favorite kind of animal. There's thousands of species, tens of thousands of species of nudibranchs, which are a type of sea slug. They're called nudibranchs because of their naked lungs. So if I take the plane of focus back out a little bit, you'll be able to see that kind of plume of dingles in the middle of its back. Those are basically gills. They're these really, it's a fine filigree of their body that extends out into the water column, allows them to exchange gases. And this one, I believe, is Hedgepeth's dorid. And of course, this is not technically a part of the plankton. This is a benthic invertebrate that's going to be found crawling along the bottom or along a dock or algae or stuff like that. But it ended up in my sample because I was towing close to a dock, and I love nudibranchs, so I'm gonna include it in here. All right, now I took that teeny nudibranch and I put it under the compound microscope, and so now we can get a closer look at it. Oh, cool. I'm guessing the red dot in the middle of it is probably, well, actually, I don't know.
A pimple? It's looking at you.
Whoa. Wait. Alright, here you can see a 6-armed brittle star. Again, not actually a plankton animal because it is benthic, it lives on the bottom. But because I went near a dock, I got it in my sample, and we're going to look at it because it's cool. You know, most echinoderms have pentafold symmetry, so 5-sided symmetry, whereas this has 6 arms, which is a little odd. I found another brittle star and another clump of bryozoans. I believe this one is a different species. You can see this one only has 5 tentacles instead of 6. So now you can see I zoomed in on this pipefish's head a little bit. Cool. You can see its little eyes looking around.